All right, ladies and gentlemen, welcome back. So we are discussing cell structure, and one of the biggest tools that we have in our arsenal as biologists to study cell structure is the microscope. So generally speaking, most cells are too small to be able to see with the naked eye, and so we utilize microscopes as a way to be able to see into that structure. Now, what kind of images we produce by these microscopes and what we want to see is driven by a handful of factors. So when we look at microscopy in general, we're really limited or driven by three qualities of an image. Its magnification, its resolution, and then its contrast. And different types of microscopy techniques, as well as different microscopes themselves, have to be chosen appropriately based on those three characteristics and based on the questions that we're asking and trying to answer. So magnification is simply how much can we zoom in? What is the ratio of the object's image compared to its real size? And every type of lens, every type of microscope is going to be limited by how much I can magnify it versus how much resolution can I get out of that. So when we talk resolution, we're talking about the minimum distance to distinguish 2 points. What's the clarity of the image? Think about a picture taken from the back of a rock concert and you try zooming in on that stage to see your favorite band, and all you're seeing is a bunch of blurry pixels because you don't have enough resolution in that lens of that camera to be able to achieve the magnification that you want and still retain the clarity. Hopefully that makes sense. So I can zoom in as much as I want, but if I lose the resolution of the image along the way, then it doesn't really provide me any benefit. So those very much go hand in hand. And then contrast is also important, especially when we're looking at images under a microscope, is to be able to see the differences in the parts of a sample. So most of our samples that we're looking at under a microscope are living things, which means they are three-dimensional. And so I need to be able to maximize the contrast to still be able to see those differences in those parts. Or once again, I lose the ability to distinguish components, and essentially I'm losing data along the way. And so that's not really helpful. So there are a lot of different types of microscopes that exist, different techniques that exist. So let's kind of take a look at just a handful of them, give you some overview so that when you come across these in college or beyond, you've at least got a sense of what they're talking about. So at this point, most of you are probably familiar with a traditional compound light microscope. These are ones that are generally readily available in middle school, high school sort of settings. Not that they aren't used in college, that is also the case, or even in research for that matter. So there's a couple of pieces here when we look at your compound light microscopes, right? We are using visual light, visible light. So usually from some sort of lamp source. If you go really old school to early microscopes, these were often mirrors reflecting sunlight up. And what that does is that light shines through a hole in your stage where your specimen is located, and then we shine that light through two lenses, right? You've got a compound light microscope as opposed to a simple light microscope, which would have a single lens. And so you've got an objective lens here, which we can rotate along that nose piece and change its magnification. And then you also have an eyepiece lens up here as well. Typically this is a 10x, and then you've got a handful of ranges down here, anywhere from 4, 10, 40, or up to 100x down here. Now remember, you have to multiply the magnification up here along with the magnification of your objective lens to get your total magnification. When we look at light microscopes, they are topped out at 1,000 times the original size. So a 10x objective in that eyepiece plus a 100x objective is the top end of a light microscope. In fact, those 100x lenses are what we call oil immersion lenses, and so you can't even use a 100x objective just straight onto a slide. You'll actually put a drop of oil between the objective and the slide in order to increase the resolution of that, because the air passing through those lenses and that light actually distorts it so much, the oil allows for a better travel of that light through it. So that's where we top out with microscopes like this. Now, once again, what are you looking for? What's your question? What are you trying to answer? Drives what kind of microscopes you use. So when we talk light microscopes, these are generally good for most subcellular structures, organelles, or I should say, is not useful for most of those substructures. Typically, the only organelles you can see in a compound light microscope are definitely your nucleus, that's usually big enough. And in a lot of cases, with plant cells, we can see the chloroplast, the central vacuole, but that pretty much tops out what we can see. So if I want to see more subcellular structure, mitochondria, Golgi, ER, that kind of thing, then I need to go to something else. The compound light microscope is not going to be able to be functional to look at most of those subcellular structures. Flip side to this is that I can look at both living and non-living things under a light microscope depending on what sort of treatment I do to it. So I can observe living things alive and functioning under a light microscope. I'm not going to necessarily have that option with every microscope choice that I have. So when we look at the microscope, light microscope, we can talk just plain, straight, bright field microscopy, which is what you're looking at here. And this is generally pretty good for looking at any sort of living cells. But if you look at it, you can see it's really challenging to get that contrast, to be able to distinguish one piece from the next. And so the other alternative to bright field is bright field with a stain. And that's what you're seeing here. These are cheek cells. And now I can definitely see that nucleus. I can start to see the cell membrane much clearer. I might be able to see some other smaller components as well. The difficulty here is that bright field staining specimen, that stain, first off, you need to choose the appropriate stain based on what you're trying to look for. Some stains will stick to certain macromolecules in cells better than others. The other piece to that is a lot of the stains will end up killing your cell. So bright field is going to be really useful if you're looking at living things, but again, the downside is the contrast. Using the stains, on the other hand, gets you the contrast, but oftentimes ends up killing your specimen. So you often end up looking at preserved cells as opposed to living ones. Okay, so what's another option? Not one that's typically readily available in a high school setting, but we can look at phase contrast microscopes. So there's a little bit more to the way that this light gets passed through the lenses. And now I can get much better contrast. Here's that cheek cell again, but I don't need a stain to do it, which means I can use those phase contrast microscopes to get the contrast that I'm desiring without having to kill my cells by the stains themselves. So I can use these as a way to increase the contrast and still look at a living thing, which is pretty cool. Differential interface contrast microscopes, similar idea. Again, now we're using different materials. It's light-ish, but you can see that there's no light passing up through the slide and through the stage at this point. These guys get us as close to a 3D image as possible. Again, I can still look at living things with this, which is pretty nice, and no stain is required as a result. But again, I get some depth out of this now, and I also continue to increase that contrast as well. But once again, they're topped out at around 1,000 times its magnification. Fluorescent microscopy, also pretty fun, interesting way to look at things. And again, typically not something that's available to high school students, but you may come across this in college, is to look at tagging specific molecules in a cell with certain fluorescent dyes or fluorescent antibodies. And so when those substances get absorbed by the cell and then we emit UV radiation as well as visible light, we can get all these crazy colors, which is pretty neat. So different stains sticking to different molecules and being able to see what they do. In some cases, depending on whether you use a dye versus an antibody, you can use fluorescent microscopy to watch living cells. In other cases, it's preserved, so it's just a matter of what sort of materials you're using to get those colors to show up and whether or not that UV radiation does enough damage fast enough to the cells that it just ends up destroying them as a result. Confocal, right? Confocal microscopes, these guys use lasers as well as some other special optics for optical sectioning. Again, these guys are fluorescently tagged. And again, depending on what sort of materials you use, sometimes you can look at living things here. Otherwise, you're looking at dead cells. But again, it gives you a lot more understanding of structure. Plus, depending on what kinds of dyes that you use, if you know what it's supposed to tag in terms of macromolecules, that also gives you some information in terms of where certain compounds might be located within a cell. So here's just our paramecia again, right? Just kind of giving you some size, ability to look at what we're dealing with here. The paramecia here are under a phase contrast microscope, so we can watch them swim and still be able to see some more of that detail because we have increased that contrast. But this is the resolution most of you are probably used to have utilized up to this point. Most of our microscopes here top out at 400x that we use with the underclassmen. So this is the top end of the microscopes most of you have probably used up to this point. So what if I want to see more? What if I want more magnification? Well, then I have to go into electron microscopy. And that then begs the question of, well, what do I want to see in more detail? Because that will determine what kind of electron microscope I choose. So these guys, pretty much collegiate level kinds of deals, they're big, they're huge. Most of them get stored in a closet, or small classroom, or something like that, because they definitely take up a lot of space. So here is where your specimen would go. This is the focuser for the electron beams, and then it's connected to a computer that will ultimately read the information and produce the image. So a transmission electron microscope will focus the beam of electrons that goes straight through the specimen. So my specimen sits here, my collection plate is down here, electron beams go through that specimen, I collect down here. It's sort of like standing up against a wall and having somebody throw a bunch of paintballs or something at you and you get that outline, right? That's kind of what this does. So what that collection plate and the computer are looking for is looking for hits of electrons versus blank spaces because that beam of electron has now hit a hard surface and not been able to penetrate. Hopefully that makes sense, and that's how the computer determines the image. Transmission electron microscopes are really good for looking at internal structure. Okay, so I can look at things from the inside. Downfall about electron microscopy across the board is that these guys A, get stuck in a vacuum, and B, often get sprayed with some sort of metal coating in order to deflect those electrons appropriately. Nothing's going to survive a vacuum. Nothing's going to survive being sprayed with a bunch of metal. So when we're looking at transmission electron microscopy or the scanning electron microscopy we're going to get to in a second, those electron microscopes require our specimens to be dead. So there is the drawback here. I can see a lot more detail, which is the benefit. My magnification goes up tremendously, but I can't look at living things. So let's go back to that paramecium a second, right? So here's what I said most of you are used to seeing. What we're going to look at in that next image is we're going to zoom in on one of these little ciliary hairs and be able to look at the internal connection. So here's that cell membrane, the ciliary hair sticks out here, and that's what you're seeing. It's just that little piece there. And this is by no means the top end of what a transmission electron microscope can do. So this one now we're looking at 14,750 times its original size. It's a huge jump. And again, it's nowhere near the top end of what we can do, but now I can see that internal structure and how it's connected, which is pretty neat. So the other option in terms of electron microscopes is your scanning electron microscope or your SEM. And again, similar setup in terms of focusing of electrons. There'd be computers here reading out data as well. But now instead of the collection plate being down here, it's up in the back here. Because what we're going to do is allow that focused beam of electrons to hit that specimen and bounce back off. Because the scanning electron microscope is going to give me detailed study of the surface of the specimen as opposed to the internal structure. So now I'm looking at how this bounces off. Again, dead things only, because I've got to stick them in a vacuum in order to make those electron beams work. And two, these guys definitely get sprayed with a metal coating to allow the deflection of those electron beams to bounce around. So definitely not something that I'm going to be able to look at in any real living detail. I can see the details, but I can't observe what they do in a functional living thing. So here's a paramecium again. Again, nowhere near the top end of this spectrum, but look at how cute and fuzzy he is. We'd never be able to see that external structure without a scanning electron microscope because everything passed through. They're thin enough to be able to see through. And so we needed a scanning electron microscope to just see how fuzzy that really is. Okay, so other techniques that we can use in terms of determining how much of something exists in a cell. I can look at it, which is great, but what if I don't have access or there's just some other reason to not be able to use those microscopes? Then we can potentially look at cell fractionation. So a cell fractionation study takes apart the cells and separates its major organelles based on size and density. We centrifuge it. So we'll take a bunch of tissue culture, right? We'll homogenize it, so we'll literally run it through a blender and chop it up. And then we will throw it through a centrifuge. And every time we centrifuge, right, centrifugation works based on density, right? And certain speeds and durations that I run those test tubes through that centrifugation process allow me to pellet out, right, or separate out on the bottom certain material. And every time I take off the lighter material, I can run it again and pellet something new. So we can get all the way down to pelleting everything as small as ribosomes if I do it enough times for long enough, which is pretty neat. So this helps not only with the biochemistry in terms of what kind of molecules are present, but also tells me what sort of concentrations of different organelles I may find in certain tissue cultures based on the concentrations that I pull out of these pellets. And so that can give me some additional information on that cell tissue's structure as well as its function based on what kind of organelles are heavily concentrated there.